Good morning, everyone. Students, we all have discussed about the various aspects of the basic immunology, right? We all have seen that how this immunological challenge is been given by the immunogen and how this immune response they act in the varying manners. Now, see, when we were talking about the applied immunology, wherever we are studying applied, so applied is the subject where which we always study after the basic knowledge. Now we have learned the basic mechanism of the immunology, right? We have seen how these immunological responses start occurring. So how the knowledge of this basic immunology we can apply. Basically, this application is based into the medical sciences, apply to understand or to study the disease. So in the applied immunology, we all are going to learn basically antigen antibody reactions. Now see, when we were talking about antigen and antibody, what we all have learned that this antigen they binds with the antibody. Now we have learned two important region. One region is present on the surface of the antigen. That particular region is known as the epitope, EPITOP, and the region which is specific for binding with the antibody is known as the paratope. Now we will have seen this epitope and paratope. They are identical or similar. Suppose say example, if your antigen's epitope region is having the V shape, so the paratope will exactly have the same V shape only. Then only they can bind perfectly. So how they bind, whether they are binding perfectly, whether or they are binding just loosely, how they bind, and after binding, what happens? They forms the agglutination. Students, you remember in one lecture I told you about the various terms: agglutination, precipitation, and the coagulation. You remember? So in that case, I told you agglutination are the molecules, or you can say the substances, which is formed because of the complex, and this complex they do not lose their properties and they remain suspended inside the solution. And when we talk about the precipitation, so precipitation something which is settling down. Okay. So after forming lots many of agglutination, and when the reaction completes, okay, when the antigen binds antibody thoroughly, and after giving the suitable type of the you can say chemical environment, they forms the precipitation. And this precipitation is very important in applied immunology. Why it is important? Because the amount of precipitin shows that how much antigen antibody reaction is being done. So we all are going to learn about all this, where this precipitation carried out and how this immunological complex are being used for the various study will be the major topic. So in general, I will just tell you what all topics we all are going to deal in this particular unit. In this unit, basically, first of all, we will be talking about what is the antigen antibody reaction under the headings of general feature of antigen antibody reaction. What are the characteristics that we all are going to learn? Thereafter, we will be talking about the precipitation reaction, immunoelectrophoresis. Now, electrophoresis we all have already learned in SYBSC, in that we have seen how electrophoresis can do. Now, in PYBSC paper 2, practical 2, semester 5, we all are going to do the electrophoresis practically. We will be performing the gel electrophoresis. Both the electrophoresis we all are going to do. Electrophoresis already we all have done in SYBSC for the separation of the protein. So we are going to do vertical electrophoresis as well as the horizontal electrophoresis in practical. Then agglutination reactions we all are going to do. Then thereafter we will be talking about the immunoassay. Immunoassay, especially we will be discussing in detail about the ELISA. Then This is all about the antigen antibody reaction. Now, vaccines and vaccination will be the major topic in this unit. Now, last unit I had just discussed a small part of the vaccine. In that, I told you that these vaccines are very important for formation of the memory cells. But in this unit, we will be talking about the principles of vaccine. What are the classification of the vaccines? Adjuvant use for human vaccines. Adjuvants we are going to learn in detail in this particular lecture only. Then vaccine against the human pathogens. And at the end of this unit, we will be talking about the transplantation, how it is related to immunology. See, in the case of the transplantation, students, we all have discussed that transplantation is one of the operation or surgery in which the transplant, or you can say it can be anything, eye cornea, it could be lung, kidney, so that particular organ is taken from the another human sibling or anyone. So anything which is coming from the outside is considered as non-self by our immune system. So how the immunological reaction happen, we will discuss in detail. So let us begin with the antigen antibody interaction. When we talk about the antigen antibody interaction, so definitely we are talking about here with the humoral immunity. Now we all have learned about the mechanism of immune system. In that we all have seen that immune system is having two action mechanism. One is the humoral immunity and another is the cell mediated immunity. When the cell mediated immunity, we all have discussed, we all have learned the role of the T cells in clearing of the clutter, or you can say the antigen, by killing them, or tumor affected cell, infectious cell, simply they are killing by producing the cytokines, or you can say the cytotoxins. But when we were discussing about the humoral immunity, that time we will have seen that immune mechanism, they design the specific antibody, right, when they are challenged, and they think like that there is a complete army of this particular antigen, and that is spreading in the entire body. So that particular time we will have seen that this immune system is challenging the B cells to prepare the same antibodies, or the antibody which is having the design of its paratope region, which is similar to the epitope region. So this particular type of mechanism is known as the humoral immune response, because I told you humoral means something which is present in the fluid. So these antibodies, they are sent inside the blood and they just roam. So for that particular testing, that how much antibodies are present, which type of test we will do, we will do the blood test. And the blood, in blood, students remember, tests, blood tests which are done to count the antibody or any type of the immunological reaction are known as the serological test. It is not there in your syllabus, but you should remember. So such type of test gives the idea that how much immune system is become activated. So this antigen antibody reaction is something which causes the humoral immunity in our body. Humoral immunity is also called as the antibody mediated the immunity. Now, how this antibody binds with the antigen? They bind the antigen with the non-covalent reaction. Now, you might be thinking that then antigen can leave off. Yes, it can, because this is the reversible, but these antibodies, they are very much firm. Why they are binding non-covalently? Then also they are firm because their sizes are identical and they are having the proper shape. Say example, you have anything which is having block ही ले लेते हैं simply. So if you will put the similar type of block, so they bind with each other covalent. non-covalently but firmly. So here also they, although they bind non-covalently, they bind very much firmly. So which type of non-covalent reaction happen here? Hydrogen bonding, ionic bonding, hydrophobic interaction, van der Waal interaction. So these are the various type of the interaction which can be seen, and we had already discussed all these reactions, type of reaction in the earlier class. Now when we talk about the antigen antibody, so what will happen, suppose now here you can see the moon shape antigen is there. So the antibody is also having the moon shape. Usually these two, both of the, all the antibodies, they have the two arm, right? We have seen that all the antibodies, they can bind to two, matlab one antibody can bind to two antigen, right? We have already learned this. So if we will be talking about the antibodies which are pentavalent, so they can bind to four, and sorry, they can bind to the 10 antigens. When we talk about the tetravalent antibodies like IgA, secretory IgA, so at single time they will be having the four paratope regions, so they can bind with the four antigens. So these are the property of the antibody. So see, as antibody, one antibody can bind to two antigens, so antigen will also have more and more epitope region. Now this is one single antigen is shown with the green circle, and this brownish shade is used for expressing its epitope region. So you can see this one antigen is having the three epitope regions. So what happened? It is having chance to bind with three paratope region of the antibody. So this is called as the avidity. We all are going to learn in detail. So that means they can bind, and because of binding of so much antigen, so much antibody, their complexes are being formed, and these immunocomplexes are known as the agglutinations. So when we talk about the physicochemical property, so antigen antibody interaction, they involve the intermolecular forces. Now what type of forces are these? Electrostatic bond, hydrogen bonding, then they can form the van der Waal forces, hydrophobic interaction. And because of this intermolecular forces, they come in close contact of antigen. Now these forces, they make the antigen and antibody stand together firmly. Now in between, when they stand together, antibody do its work. Now antibodies are not the simple molecule, we are just doing the opsonization of the antigen. They are going to react with this antigen, they are suffocating the antigen, and antigen is something the life body, and because of this suffocation, the antigen, because of this binding, this suffocation, the antigen will die off. That we have already learned. So this stability of antigen antibody interaction, we can say it's totally depend on the good fit between the antigenic determinant and antibody combination site, that is epitope and paratope region. So if both the things are good fit, then only they have the stronger reaction. Multiple intermolecular forces can be attract at one time. Now it is not like that they will be having only hydrogen bonding, they will be having only wonderful forces at the same time. They may have the hydrophobic interaction, they may have the van der Waal forces, they may have the electrostatic forces. All the forces can act together and keep this antigen antibody binding together very firmly and very tightly. Now the another thing is affinity. Affinity means, yes, what do you mean by affinity? It is a general term, students. In general, we know that affinity is the attraction, right? And the opposite word of affinity is phobia, phobic, right? Phobia means going away from the particular thing. So affinity, when we talk about, is the attraction. So there is epitope region is attracted by the paratope region. So if they have the low affinity antibody, so they will bind weakly. But if the antibody, now see why this low affinity will occur, because antigen is different. Okay, antigen is different, so definitely their epitope region will also be different, but it is little bit match type. So they may bind to the antibody but not that firmly, but the antibody which is designed for the specific antigen, and they have the perfect paratope and epitope binding, so that particular time they will be having the high affinity. Now another term is the avidity, not specificity. Another term is the avidity. Now what is this avidity? As I told you that antibodies, they are multivalent, and most, because of that, the most of the antigen also have the multiple antigenic determinants. They may have the more than two epitope, so they combine in the varying proportion. Suppose this is only one antigen, and this antigen is binding from the, now see, this particular antigen is having two antibodies bound together. So this is known as the avidity. That means it is, you can say, greater than the affinity. So avidity is binding strength of such and multivalent antibodies and antigen having the multiple epitope. When we talk about inside the biological system, and avidity of an antibody molecule is always better indicator of its binding capacity than affinity. Suppose if this is the affinity, so this is avidity. Avidity is what? One antigen is bound on the multiple side, multiple paratopic region. So we can say avidity of an antigen antibody reaction is totally depend on the valences of both antigenic side and the antibiotic side. So their binding is the sum total of individual affinities. So that means this particular antigen is bound with, this is pentameric your IgM. Now we all know that there is only one antibody which can form the pentamer. So this IgM is having the maximum avidity. Why? Because it is having the 10 antigen binding site, or you can say the 10 antigen binding vacancies. They can bind to the 10 epitope region. So here they have the maximum avidity. So it may be a chance that it can bind to all of its 10 epitope, paratopic region to the epitope. So this all the affinity is what? Affinity is the interaction between one antigenic site, that is one epitope, to the one paratope. Okay, so always avidity is greater than the affinity. Now next thing, when we talk about the specificity, so we have already learned that antigen antibody reaction, they are highly specific. Okay, the ability of binding site, that is paratope of an individual or population of antibody react with one antigenic determinant, this is called as the specificity. That specifically they will bind to the one particular site only. Now antigen antibody can cross react also. This cross reactivity may sometimes cause disease in host, and it may lead to the false of diagnosis. That means they may start doing the autoimmune reaction. Now when we talk about the cross reactivity, so cross reactivity is something that antigen or one type of paratope region which is formed for one type of antigen can cross react with the another antibody. That means one individual has produced the immunity, and for one antigen, say example, we will take here example of the COVID. One person affected with the COVID alpha version, okay, and thereafter the person's sera is been given to the another person who has affected with the beta. Now delta version is present, but we will take just examples, okay, but that is affecting, okay. So for that particular reason, means they may not have that perfect binding, they may bind bit loosely, but they can bind and they can kill the COVID. Okay, so this is called as the cross reactivity. This was a basic concept considered, and COVID vaccine has been designed. Although they may not, whatever antibodies are formed after that taking vaccine may not be exactly perfectly matching. People will say, "Na, now the Delta 4 variant is there, strain has been changed, then we will get the disease." So yeah, strain is changed. You may have the different type of antibody, but these antibodies can cross react. So this is called as the cross reactivity. Although whatever antigen antibody reaction, they are highly specific. Sometimes whatever antibody elicited by antigen, they may can cross react with unrelated antigen. So this cross reactivity occurs if two different antigen will have the identical or very similar epitope. So it is been expected that whatever epitope will form, that epitope, although their versions are different, their strains are different, that but this epitopic region, they may have some similarity. So the affinity of an antibody may cross react, and this may usually less, means the way that particular true virus against which this antibody is been formed is having the affinity, this particular strain may not show. But antisera, whatever containing the polyanticlonal antibodies, are been used for treatment of various diseases, and it has been proved that antibodies cross react with antigens that are partially related to antigen used as immunogen. So this is all because of the presence of some common epitopes with similar structural configuration. So this is all about the features of antigen antibody reaction. So we have learned that antigen, they have the specific site, that particular site. Now see, this particular site will not be harmed by antibody, because this is not identical, or this is the area which is not understood by the antibody. Antibody can bind to the area to which it identifies, that is the specificity of the antibody. So specifically, antibody will bind to the area where the antigenic determinant, that is epitope, is present. And here also we need to remember the antibody will not bind to any of its site, it will bind to the paratopic region only. So what is the affinity of antigen antibody reaction? Affinity is that the epitope region have the attraction towards the antibody. And what is the avidity? Avidity means utilization of maximum paratopic region of an antibody complex. So if it is a monomer, or you can say a single molecule of antibody, so it is having affinity to bind with one of its side, and avidity is what it utilizing its both side. And what is the cross reactivity? Cross reactivity means the similar epitope of irrelated or unrelated antigens binding with another type of antibody. So this is called as the anti-serotherapy. That means they are using the serum of already cured people, and they are using it on another people. So that is called as the use of the anti-sera. Plasma can also be in somewhere, it is also related, that they are using the cured plasma of the people, and they are using it for the purpose of the treatment. So this is all about the general features of antigen antibody reaction. Any doubt? Okay, we'll stop for this note.